So there are three types of muscles in the body: the cardiac, the skeletal, and the smooth muscle. So let's talk about the skeletal muscle. Skeletal muscles have stripes, so they are called striated muscles. Skeletal muscles are completely independent of each other, meaning that they are isolated from one another. So for example, if I were to stimulate one skeletal muscle, only that muscle would contract and nothing else would happen to the neighboring muscle cells. Skeletal muscles do contract in the absence of nerve activity. And lastly, skeletal muscles are voluntary except for the diaphragm. Cardiac muscles also have striations and they are highly interconnected like a lattice. Cardiac muscle cells are really special because they're connected to each other by intercalated discs. And you can see those right here. And these intercalated discs contain a lot of electrical synapses, and they're heavily connected. Cardiac muscle cells form a functional syncytium, meaning that if you stimulate one of these muscle cells, then the entire thing will contract and the impulse goes all the way throughout the muscle cell. So cardiac muscle cells will contract in the absence of nerve activity because the presence of the pacemaker. Pacemaker cells in the heart are actually muscle cells that are modified to work like a generator of electrical current. And of course, the cardiac muscles are involuntary. For smooth muscles, they have no striations and they're super tiny. The smooth muscle cells can either be functionally syncytial or independent from each other. And in cases that the muscles are independent from each other, they would need nerve activity to be able to contract. So smooth muscles are generally involuntary except for the bladder. Now let's take a look at muscle cells more in detail. The neuromuscular junction is what drives the muscular activity. So we have acetylcholine receptors right here at the postsynaptic membrane. The EPP will generate an action potential over here, which normally lasts longer than that of the muscle cell, and the speed of conduction of that action potential along the membrane is also going to be much slower than that of the nerve cells. And just remember that the function of the action potential in the muscle cells is to depolarize the membrane so as to start muscle contraction. If you take a cross-section of a muscle cell, you will see that there's a lot of fascicles which contain the muscle cells. The epimysin, which is right here, the epimysin is a white sheath of fibrous elastic tissue surrounding a muscle, and within the muscle, the fascicle is also surrounded by a membrane called the perimysin, and you can see that right here. So here's the epimysin, and then there's the perimysin, and then lastly the endomysin is the membrane that surrounds each muscle fiber. So for each muscle cell, there is a tiny cell that is between the membrane of the muscle and the connective tissue called the satellite cell. And these satellite cells right here are basically stem cells. The satellite cells are able to convert themselves into muscle cells if needed, and they have very little cytoplasm. So the muscle cells in their membranes are going to bunch together at the end like this and fuse into the tendon, which inserts itself into the bone. As you can see right here, muscle cells have more than one nuclei because of how they are formed. Each muscle fiber is formed by a large number of myocytes which fuse together, bringing their own nucleus. and the result is a multinucleated muscle cell. So in the next picture below, we can see that each muscle cell has a large number of small fibers called myofibrils. Within the myofibril, there are thick filaments called the myosin, and then there are thin filaments called actin. The myosin and actin are arranged in a way that parts of the thin and thick filaments overlap, resulting in areas of dark and light bands, and this is the cause for striations. So right here, this blue line is called the Z-disc. This is where the fibrils are actually attached. And the area in between the two Z-discs, this is called the sarcomere, the unit of contractile mechanism for the muscle. So the place where only actin filaments exist is actually going to be called the I band, right here. And as you can see, it's really light. And then the dark bands are called the A band. This is where the actin and the myosin overlap each other. And in the middle of this dark band, we have the A band. And this is where, we have the H zone. And this is made up of only the myosin filaments. And in the middle of the H zone, we have a line called the M line, right there. So when the muscle cell contracts, the two Z lines are going to go towards each other, shortening the muscle cell. The myosin filament is made up of thousands of molecules, and from each myosin molecule, a projection comes out and away from the filament, and these projections are called cross bridges. Each myosin molecule is attached to the Z line with a spring-like molecule called titin, right here, or connectin. The actin filaments, which are much thinner than myosin, are attached directly to the Z line through a molecule called actinin, right here. Desmin binds to the Z line, to the sarcolemma from the inside, and dystrophin binds the actin filament to the sarcolemma. The sarcoplasmic reticulum is the blue-like net thing, which is covered by the sarcolemma. The transverse tubule, seen right here, is part of the sarcolemma that goes deep into the muscle fiber to bring the action potential to the interior of the muscle fiber. So each transverse tubule is going to be surrounded by part of the sarcoplasmic reticulum, and this is called the terminal cisternae. So right here, this is the terminal cisternae, and the transverse tubule that is surrounded by both the terminal cisternae, this is going to be called the triad. So 1, 2, 3, it's going to be a triad. And the entire system of nets and tubes is called the sarcotubular system. So how does contraction take place? An action potential comes down from the neuron and depolarizes the terminal, which releases acetylcholine. Acetylcholine will generate an EPP, giving rise to an action potential, which travels all the way through the membrane and dips into the transverse tubule. And this is going to depolarize the membrane, causing the opening of calcium channels in the cisternae. Calcium is then going to be released in the cytoplasm over the actin and the myosin filaments. The terminal cisternae has a large amount of calcium storage and a protein called calcestrin. The protein helps hold calcium in the cisterna of the sarcoplasmic reticulum after a muscle contraction, even though the concentration of calcium in the sarcoplasmic reticulum is much higher than in the cytosol. So each myosin molecule is composed of 2 heavy myosin molecules and 4 light myosin molecules. The 2 heavy myosin molecules form a double helix at the ends right here, and they become globular in structure right there. The globular structures form the cross bridges. So the light molecules are going to wrap around the neck of the heavy molecules, and hinges next to the myosin head allow it to move. Actin molecules are more complex and composed of 3 different types of molecules. The F-actin strand right here have red globular structures called the G-actin molecules, forming a double helix. So tropomyosin is a double helix of thin molecules that wrap around the G-actin loosely, so that it covers all of the actin sites of the G-actin. So this is the active sites right here of the G-actin, and tropomyosin is going to cover that very loosely. So troponin attaches to tropomyosin in regular increments, and it has 3 different globular molecules, one with an affinity to tropomyosin, one with an affinity to actin, and one with an affinity to calcium. So at rest, tropomyosin and troponin cover all of the active sites of the G-actin, which is why they are called relaxing proteins. So when calcium is released in the cytoplasm, it will interact with troponin. A conformational change occurs, causing the tropomyosin to move away from the active site of the G-actin, and the active site will then attach to the myosin head, which moves, making the sarcomere shorter and shorter, and this is going to be called the sliding filament mechanism. So notice that in order for the muscle system to engage the myosin head with the actin, you really need ATP. So at rest, the myosin head is cocked and ready with ADP, allowing the head to interact with troponin. The myosin head detaches from the troponin only when ATP comes around to the head. The head of the myosin has ATPase activity, which splits ATP into ADP. So when there is not enough ATP, then the head will continue to be attached onto the troponin, and the muscle keeps contracting. And an example of this would kind of be like rigor mortis. So the cell cannot do anything without calcium. Calcium inside the cell is supplied almost entirely from the sarcoplasmic reticulum, or more specifically, the terminal cisternae. So the DHPR is really nothing more than a calcium channel in the membrane of the transverse tubule. Calcium that enters through the DHPR, seen right here, will pass by the area between the transverse tubule and the terminal cisternae, right here, and it's going to interact with the ryanodine receptors right there. This is another calcium channel. So this release of calcium is going to cause the ryanodine receptors to release calcium into the cytoplasm. So basically, the release of calcium causes more release of calcium. So looks like that is all. I'll see you guys next time.